Okay, welcome to today's Kirby seminar, and I'd like to start by acknowledging the Bidjigal people and that we're meeting on their unceded lands. I'd like to pay my respects to elders past and present, and extend that respect to any Aboriginal and Torres Strait Islanders that are joining us today. So today I'm very excited to introduce Professor Paul Klenerman, who's the Sidney Truelove Professor of Gastroenterology at the Nuffield Department of Medicine in Oxford. So Paul helped train me. I met him when I was a postdoc in Oxford. We were postdocs together, and he's helped train a number of people in this room, including Chan and Sarah. So the Kirby has a great debt to Paul, and Paul has done extraordinary work over many decades now, initially in the context of immunovirology, and initially in HIV, in various models of viral infections, and also in hepatitis C. And today he's got a very ochre title of MAIT cells and their mates. So Paul, over to you.
Thanks. So yeah, I was going to talk about MAIT cells because I think they're quite underrated, and I've been working on this area for a while. And not just MAITs, just to put the unconventional cells a little bit more into context, and largely in the context of infectious diseases and vaccination. So hopefully can right the sort of wrong where they're largely ignored. Yeah, so let's think about these cells. This is a sort of general model that we've been sort of using to try and understand what they're for. So this sort of cell that we're looking at is different from a conventional cell. So conventional T cells recognize MHC peptides. This is all very well worked out. Very specific recognition. And they tend to recognize pathogens. So essentially their job is very episodic. Once a pathogen is seen, it will respond, and then it will go back into a kind of memory phase, or whatever the if it's an intermittent challenge, it will go into deep memory. If it's a chronic infection, then they will remain activated. But it's usually a sort of hit and run process. So the specific triggering is well understood, and you'll find them largely enriched in lymphoid organs, although, as everybody knows now, there's quite a lot of memory for conventional T cells, and thinking about CD8s here really, out in tissues, as tissue resident memory. And then their functions tend to also be relatively specific. So they're highly specialized, I guess, is the main point. Whereas if you think about cells which are responding to commensals, their job's quite different. So they might have a T cell receptor that recognizes something that's quite common, and that might well be seen every day if you're at barrier tissue. And so in order to modulate this, functionally, these cells have evolved alternative ways of sensing their environment so that they are not triggered continuously. And that's really a high dependency on innate sensing. So it tends to be the case that cells which are recognizing ligands which are not peptides but are derived from common cues that derive from commensals, and I'll show you what MAITs recognize in a minute. They tend to be much more innate-like. It's not a one-to-one match, but these things tend to go together. So another feature of these cells, just for sort of context, is that with this triggering, they have to make a decision as to whether the commensal is just doing its thing and is in the right place, and really no requirement for a MAIT response, versus a more aggressive response if the commensal essentially becomes invasive and is in the wrong place. And so they have essentially, what that means is that they've got a wider suite of functions, and those functions include something that's much more homeostatic. So tissue repair, as I'll show you. So they're one of the really interesting features of these cells, and this is, I think, probably true across the board, is that they can flip and flop between something that looks much more like a conventional cell killing and interferon gamma and inflammation, and something that looks much kinder, essentially, which is tissue repair. And the other thing just to mention is that the unconventional T cells tend to recognize MHC1B molecules, which are, according to some reports, evolutionarily older than the more diverse 1A molecules, which for example, a CD8 cell, a conventional CD8 cell response. So in fact, what you call conventional and what you call unconventional just depends on how they were discovered rather than their actual evolution. So one prediction from this model, which I'll come back to later in the talk, is that if you are a conventional T cell with a peptide-specific response, but you happen to recognize commensals, then you might expect some of those cells to acquire a model that looks more like the unconventional ones, because they are getting cues every day and they need to adapt their behavior to something that's a little bit more continuous in terms of T cell activation. So MAITs were defined in the 90s really by their T cell receptor. So the original studies didn't really look at their phenotype at all in any detail, but this one particular T cell receptor in humans, V alpha 7.2, G alpha 33, came out from studies from Steve Porcelli and then Olivier Lance. And over the next 10, 20 years, the restriction element was discovered, which is MR1, which is an MHC related molecule. So it's a 1B molecule, which is incredibly well conserved between humans and mice, and within humans. There's a very limited amount of polymorphism. The other feature, which is how we got into this, is that these cells tend to be also high expressors of molecules such as CD161, which is an NK-associated molecule. And CD161 just happens to be a useful marker because it's very highly abundant. But there are quite a number of different molecules you could use. Essentially, their phenotype is quite distinct. So if we took blood from any of you, we would find a population of MAIT cells in your, this is on CD8s, that are high in CD161 and express V alpha 7.2. And it's a pretty decent chunk of your CD8s. It's often around 10%, can be a lot higher. And one thing that slightly bugs me is if you do studies of CD8 T cells and don't take into account that you're including MAITs, you can actually get quite a misleading assessment of genes, particularly the genes which are not expressed very well in a typical CD8 T cell, like raw gamma and IL-17 production, but are highly expressed in MAIT cells. So I think people, even if you just want to exclude them from your studies, it's worth knowing about these cells. So there are, of course, other cells which express V alpha 7.2, which are nothing to do with MAIT cells. It's just part of their T cell receptor. So the actual ligand for MAIT cells that's bound by MR1 is a molecule called 5-OPIU. So if you make a tetramer, as was done by the Melbourne groups, that contains MR1 as the presenting molecule and 5-OPIU as the ligand, you can get exactly the same thing. And 5-OPIU is a really interesting molecule. So this is made by microbes, typically bacteria, but also some yeasts. And almost all bacteria, with a few exceptions like group A strep, will tend to make this as they make riboflavin. So really what this molecule is telling you is that a microbe is there and it's metabolically active. So that's basically the exact same population. It's just that essentially all the cells that express this V alpha 7.2 G alpha 33 are recognizing this. There are small exceptions, but it's a pretty much one-to-one match. And the reason that we're really interested in this is that when we started to look in hepatitis in livers, these cells are really enriched in the liver. So it can be up to 50%. That typically be more like 20% of the CD8s in the liver, but 20 or 30% would regularly be MAIT cells. So if you're thinking about the specificity of cells in your liver amongst your CD8s, this is by far the most common specificity. And these cells in the liver, they're not, I had expected when we did these analyses that you'd find them sort of concentrated around the portal tract so that they could recognize microbes that were coming in, or there'd be some sense to it, but actually they're really scattered around. So they're not really close to the portal tracts at all. They're sort of scattered through the parenchyma as sort of single cells. So I think this is kind of an important feature. So if we think about how the cells are triggered, they don't have a lot of other cells around them that are going to regulate them and tell them what to do. They have to make the decisions themselves. And I think that's one of the reasons that they have adapted this kind of slightly curious way of getting triggered, because they have to know whether the thing that they're dealing with is dangerous or not. So this is the kind of area we worked on, which is more like the function of the cells and how they kind of use these little modules. So one of the strong features of MAITs amongst human T cells, but particularly amongst human CD8s, is a very strong type 17 bias. And amongst cells in the blood, these are the cells that express the highest levels of IL-23 receptor. And lots of cells can express it in tissue. And one way of looking at MAIT cells, in fact, is that they're kind of already got many of the features that a tissue adapted cell has acquired within tissue. It's all preset within the MAIT cell. So they've certainly got so this is one of the standout features, although as I'll show you, they tend not to use it very much, but they're programmed to do it. Obviously the TCR, and they've got very strong, additionally, features of type 1 immunity. So it's a completely mixed phenotype, and they make lots of interferon gamma. They can kill. I'm not going to really talk about this, but they can upregulate granzyme B and kill very well. They have strong innate features, including these molecules like CD161. They'll express CD56 as well in some cases. And they express lots of chemokine receptors, including CXCR6, which is probably the reason they're found in the liver, but plenty of other ones which will take them to tissues. And then, so this diagram I'm going to spend a little bit of time kind of referring to. And so this is what I mean by the kind of flip-flop mechanisms of activation. So we talked about the TCR. So as if there are microbes that are making riboflavin, that can be presented, but MR1 is quite ubiquitously expressed, although how it's loaded is a bit of a mystery, but nevertheless, somehow the ligand gets onto MR1 and you get TCR signals. But if you just get TCR signals, you get quite a limited set of functions. And to summarize those, they tend to be more of the tissue repair homeostasis ones. I'll show you an example later in the talk. But it's a partial response. You can get some interferon gamma. You can get a little bit of everything, but it's less of a kick, and it's more of this sort of these slightly unusual molecules like furin. And then on the other side, if you get inflammatory signals on their own, and this is more like an NK cell, if you get IL-12, IL-18, IL-15, and a lot of, importantly, interferon alpha, and as I'll show you, TNF, they can get triggered independently. So in a viral infection where there's no vitamin B being made or anything, and you can show this is totally independent of their TCR, they will also trigger off, and they'll create a slightly different set of responses dominated by interferon gamma, but again, quite a broad range of responses. And sometimes this can be beneficial, and sometimes it can be detrimental, and depends really, like any T cell response, about timing and sort of magnitude and appropriateness. But certainly it's a very strong feature of their biology. And actually, even in the presence of, there's a nice paper on Klebsiella infection, and even in which does make the right metabolite, but even there, actually the triggering in vivo seems to be much more due to the cytokines rather than the ligand. And then finally, if you get both bits together, then you get maximal responses, and you can kick really a range of things into gear. And that can be protective. Again, it could be pathogenic, depending on the context. So we've done some, like everybody, sort of single cell work. And so I'll just show you a little bit more what I mean in just using some of the single cell work to describe this. And I like this assay. So Lucy Garner in my lab set this up. So what she did was she took MAIT cells, sorted them from blood, and then triggered them, either left them unstimulated or triggered them just with a TCR trigger using the ligand and MR1, or just a cytokine trigger just using IL-12 and IL-18. And then she left them for a bit and then did a 10x protocol and looked at what had happened. And you get this nice sort of wishbone effect. So the cytokine stimulation sort of goes up this trajectory, and the TCR stimulation goes down that trajectory. So it's not to say that there aren't shared genes at all, but it's nice to see that they're kind of distinct profiles. And if you just trigger them any old way, you don't get the same thing. You get different programs. If you look in a bit more detail of what that means, so if you look at the cytokine stimulation, for example, the interferon gamma gene expression. So this is at the tip of this are the cells that are maximally stimulated, so that they're making loads of interferon gamma, whereas you get much less of that in the TCR triggered, whereas TNF is a little bit more TCR triggered. Granzyme is cytokine triggered. R26, which is a molecule that we don't really understand very well what it does, is much more of a cytokine triggered molecule. And then there's some things that are downregulated, including CD161, that's a gene for it. So as you stimulate the cells, it tends to get a little bit downregulated, which can be a nuisance if you're trying to track the cells in vivo, but it's usually manageable. So you can sort of dissect that out, and I won't go through the whole paper. It's published if people are interested in the details, but you can sort of track individual clones along these trajectories. So individual clones traverse these trajectories at different speeds, and it depends which trajectory they're going down. So some of them are more easily stimulated by cytokines, and some are more easily stimulated by TCR. And you can separate out the clones because although the alpha chain is highly conserved, the beta chains are quite variable. So at a single cell level, you can sort of assess the functionality of these clonotypes. So I sort of, a part of the reason for putting this up is I think if you're doing studies of these MAIT cells are really easy because there are lots of them and we know the ligand. But if you're looking at HIV or CMV or something else, you could do the same experiment. You might just need to work a bit harder to find the original population, but you could actually probe their function really at a clonal level quite deeply. So one of the unusual things that we found, and this was a kind of reviewer's comment, was we didn't find in these studies where we just given one TCR or cytokine, we didn't find any R17. And so, well, what's going on? And the reason is that you need to do a bit more. So you need to give both together. And then you can, so this is the same wishbone. So it's cytokine, and all the TCR stimulated cells are sort of jumped across. We haven't got any in the middle, but that's a sort of wishbone. And then the red group are the double stimulated cells. So they're kind of fitted nicely in the middle. But there's this little kind of island on the outside that you can see. And that's a later time point. We ran out of money to do all the different things for this. But interestingly, the R17 cells form their own little group here, little cluster. And they're not that many genes in the cluster. It's quite an interesting group. But what seems to be happening is that cells, and any cell can enter that. It's not a unique feature. There's no pre-existing R17 type of MAIT cell. They can all do it. And this is a mixture of different TCRs. But it just seems to take time. So if you look at the epigenome, the R17 locus is actually relatively less open than others, and it probably just needs to unwind a bit. But it seems like essentially a little bit of a protective feature that they won't start to make R17 until they've really had a lot of TCR and cytokine triggering. So that's essentially their sort of mechanism for controlling that. So I'm going to talk about viruses. I'm going to give you an example of some examples of viruses giving a kind of injury and triggering MAITs and what they do. And then I'm going to talk about this side. So a while back, we showed that this was a little bit against the grain because the TCR for the MAIT, as you can see, is highly bacterially sensitive, and viruses can't generate that ligand. So people said, well, they can't respond to viruses. But if you actually look at patients, they're responding incredibly well to viruses. So this is acute dengue. These are pediatric samples. This is chronic hep C. These are patients that had cleared a long time ago. And these are patients who'd either have or have had chronic infection. This is a small group of acute influenza, and the group in Melbourne did a lot more. And in COVID, you get exactly the same thing. I'm not going to show you the COVID data, but you could imagine what it would look like. So what we're looking at here is granzyme B, which is a good marker of the activation. You can use CD69 as another marker. So in all these contexts, there's quite strong activation of MAIT cells. And then what we showed a little while back with Jim McCluskey's group was that if you take MAIT cells away, and this isn't a mouse challenge. So the question was like, well, okay, so they get activated, but so what? But it's important because if you take them away, and this is a mouse that lacks ML1, and what that means is that they can't select MAIT cells in the thymus, so they lack MAIT cells. But they're otherwise, in a mouse, it's very, very small population of cells in MAIT cell by comparison with the humans, about 100-fold lower. They tend to use iNKT cells instead of MAIT cells, so they sort of flip-flop. But so these mice don't have MAIT cells. And these mice we put MAIT cells back into, but they can only respond in a cytokine-dependent way because they haven't got any MR1. So what happens with the mouse, if you give them a decent dose of this severe flu, PR8, they will lose weight and then they will kind of recover a bit. And if you get the dose right, they'll just skim past this 20% line, which is where essentially you have to terminate the experiment. And that's essentially a fatal outcome for the mouse. So, and you can see it's not that big a difference. We've reproduced this again recently, and it's pretty much the same. The weight loss isn't hugely different, but they're tending, what's happening in the absence of MR1 is that they're tending to just cross the line and not really scoot past. So they're just losing a little bit too much weight. And when you translate that into survival, it's quite a big difference. So most of the wild type mice will survive, and most of the MR1 knockout mice will die. And then if you put MAITs back in, you can recover some of this. So that was really important, actually, in old data, but I really think it's an important feature because up to that point, we hadn't really sort of had any really solid evidence that this mechanism of activating MAIT cells had any role at all. I'm not going to show the data, but I should say it's not to do with the viral load. The viral loads are very similar in the wild type and knockout mice. It's probably to do with the recruitment of other cells. So everything is just a little bit delayed in the MR1 knockout mice. And we don't really know 100% know the mechanism that's behind this. We're still looking at that, but it's probably to do with recruitment of other cells that have got a kind of overall protective response. It's quite complicated in the end, but fundamentally it's important. I thought I'd show this data from the Chan and Prab and Tony helped with this paper. In chronic HIV, or early HIV, you can see, again, all these, that's the sort of activation. This is PD1, granzyme, TIM3. If you look at granzyme B, which is equivalent to what I just showed you, there's actually quite a lot of, BL is baseline, and that's after treatment. There's quite a lot of activation of the MAITs in HIV. And the same question arises, and obviously we can't do an animal experiment to show whether this is important, but there is some in vitro. It's not I wouldn't say it was sort of, they're the cause of HIV protection, but they're at least a component of it, I think. So if you look at the activation, then if you give E. coli, you can block that very well with anti-cytokines, these antibodies to the cytokines, so you can sort of block that well. And similarly with HIV, you can trigger the MAITs in vitro and you can block it with the cytokines. So, and then the question really arises is anything that the MAIT cell makes suppressive? Is it doing anything? And you can show that if you take MAIT cell supernatants, which have been triggered by HIV, it will suppress in this reporter assay HIV replication. And then if you block chemokines, you can block the inhibition. So that amongst all the things that they do make some antiviral chemokines, and they will make some interferons. And I think there's a little bit more to say in this, but essentially that's where we got to with that paper, which was heroically finished during the pandemic by Chan and Kev. So what I was going to talk about for a few minutes was about vaccines, because we published a paper showing that MAIT cells could be triggered by adenoviral vectors, everybody's favorite vaccine platform. Well, at the time, ChAdOx, when we did the studies, nobody had been given any ChAdOx outside a clinical trial. And now it's been given to millions of people. And what you find, and I'm summarizing a lot of data, is that ChAdOx will trigger PDCs really strongly, and they will make interferon alpha, which we know is a MAIT cell trigger. It'll trigger monocytes to make IL-18, which we know is a MAIT cell trigger. And what we found, interestingly, is that interferon alpha triggers monocytes to make TNF, which I had not really known, and it's apparently deeply buried in the sort of 1980s literature. It's quite hard to find, but it's quite potent. If you drop interferon alpha onto monocytes, they'll pour out TNF. And the TNF is important because if you block it, you can't get full activation of the MAITs. So it was this combination. So that was the paper. And again, using a mouse model, this is a vaccine that is basically ignore all the kind of bits and pieces. It's a Hep C vaccine that was made with ChAdOx. And again, in the absence of MAIT cells, you don't get very good immunogenicity. It's not the world's strongest immunogen, this vaccine, compared to the COVID one. You get probably 10 times as much T cell response from the COVID vaccine in a mouse, but you get the same phenotype with COVID, that you reduce the amount of T cell activation. So that's the number of gamma secreting cells in a regular mouse by ICS, and then in the absence of MAITs, it's actually quite significantly suppressed. So we don't really 100% know all the mechanisms, and there are not that many MAIT cells in a mouse lymph node, which is probably where this is all happening, but there seem to be enough to make the difference, and something to do with recruitment and activation. So I thought I'd talk about this work by my current clinical lecturer, Ali Amini. And so it was done during the pandemic, but I thought you'd be interested. Basically, it's a study where we captured people who were going through the first wave of vaccination. So naive, some of the people included had previous COVID, but largely naive, captured it 1 day after vaccination and 1 day after boosting. And then we had 2 groups in the UK of people who received Pfizer vaccination. The standard 3 to 4 weeks, as was suggested by the company, and then the UK version of the vaccine interval, which was about 8 to 12 weeks. And that was done because we wanted to maximize the number of people that got the first dose as early as possible. And there was some logic that actually, if you extended the dosing interval, you might get better antibody responses, which turns out to be true. Anyway, so there's a lot of stuff that we did, and that's why it's taken a little while to kind of work through. But I'll show you some of the data because it's quite interesting. So what happens with the MAITs is, if you look at the ChAdOx, as you'd expect, you get that big burst of cytokines, and then 1 day later you get a big burst of MAIT cell activation. And as I showed you, that supports the production of T cells at least. So we know that much. But with the boost, you get much less. So that's kind of what's shown here. It's MAIT cell activation first, and then not much less second time. And but it's the opposite with the mRNA vaccine. You get hardly any. This is the p-values there, but as you can see, it's hardly any MAIT cell activation by comparison with the RNA vaccine, which is what you'd expect because it's been engineered to basically remove all its innate stimulation. It's had the pseudouridines put in to prevent triggering. And but having said that, the second time around, exact same vaccine, you get a big activation, and so that's really what it shows. And it's not just MAITs, but the MAITs are probably the easiest one to see. The NKT cells do the same thing, but there are not very many of them. And if you look at the cytokines that go with that, it's basically a mirror image. So the ChAdOx is the gray and the Pfizer is the purple, and we're comparing prime and boost. So with ChAdOx, you get lots of interferon gamma, particularly at prime, and less at boost. And it's a reverse with the this is looking at circulating cytokines, and the interferon alpha is really just a feature, as far as we can tell, of the ChAdOx rather than the Pfizer. You can get transcriptionally, you can get a signal for type 1 interferon, but there's these curves are completely flat. So there's a big difference in gamma. And so we're just obviously one reason that you might expect that the ChAdOx has a lower response at boost, or lower essentially innate response at boost, is because of induced antibodies. And that's what I'll just show you. And certainly if you just add the antibodies from, or sera basically, from the vaccinated patients into an in vitro assay where you just dump the vaccine into PBMCs and look at infection, you can see this little bit of the graph is the infected cells. So if you use free vaccine sera, or people who've been vaccinated with the Pfizer vaccine, there's plenty of infection. If you take sera from people who've had ChAdOx, it's pretty much killed off. And that's what the results show. And therefore, it fits with the the activation of the MAIT cells. So there's little infection, so therefore there's very little triggering through the mechanisms I showed you. So that's fairly straightforward. So you get lots of innate activation with the first dose of the ChAdOx, and much less with the second, because it's all been neutralized. But what's going on with the Pfizer, because first time around, there's very little activation. So how come they're getting all this signaling second time around? And I think the reason is probably interferon gamma. So what is interesting about the MAIT cells, you can see, is that they express interferon gamma receptor. And they do that much more strongly than regular cells. So these are regular CD4s and 8s, and MAITs express more than NK cells and more than most things. So they are expressing gamma. And I think this is an interesting experiment. So if you just take somebody who's been immunized or infected, but with SARS-CoV-2, they'll have T cells. And if you do like a standard T cell assay, which I'm sure lots of you in the room are kind of doing at the moment, and look for interferon gamma secreting cells, and then gate on MAITs and NKTs and gamma deltas, you don't get much response to spike peptide. You can do this with vaccine, but if you put spike peptide in with PBMCs, most of the cells that will fire off are CD4s and CD8s. For some reason, the CD4s seem to dominate, and that's what you'd get on your ICS. So that's at 8 hours. If you look at 24 hours, you get a completely different response. So the MAITs have really kicked off. And a lot of the cells that are making gamma at that time point are actually your unconventional cells. I mean, you still got some CD4 and CD8 responses, but all they can do is keep on making gamma. That population is whatever your memory pool is. Whereas this population of MAIT cells is your entire population of MAIT cells, which I've already said is like maybe 10% of your CD8. So it's going to dwarf the other population. So I think this is a really interesting result, because we do these assays all the time. We do ELISpots, we do ICS, do, fax-based assays. And somehow we've sort of ignored the fact that a lot of the activation potentially can be through these bystander mechanisms. And a lot of it will depend on what time point you use. And the amount of gamma that you see totally is very correlated with the amount of MAITs. So although the MAITs are not responding to the peptide directly, they're responding very strongly indirectly. And I think that's what's happening. We can show that that's gamma dependent, because if you do the same assay with your spike peptides and then block with the anti-interferon gamma receptor, that's anti-CD119, you can kill off this entire response. And that's basically the facts. That's your MAITs with the isotype, and that's the MAITs with the... So it's all really gamma dependent. Gamma on its own is not going to do this. It needs to be combinatorial with other cytokines, but nevertheless, gamma seems to be the dominant thing. And just to kind of make it a little bit more interesting / relevant, if you go back to this long and short dosing interval, which I think it's only the UK and Canada used it, the people with the long interval, they tended to have less interferon gamma found at their boost, and they also tended to have slightly better antibodies. And if you look down this sort of bit of the correlation curve, you can see there's more antibodies with a longer interval, generally less cytokines. And I mean, most of the stuff about to do with immunogenicity is kind of correlative. So we haven't really shown exactly how this gamma is impacting on immunogenicity, but I think the reactogenicity might be really important, because that ultimately is something that really limits the use of these vaccines. So if you just bear with me a second. So if you take the short interval, as I've just shown you, this is from the Comcov trial, and it's a cartoon of reactogenicity. So Comcov was a really nice trial where they mixed and matched adenos, RNA, etc., at different timings and it was a really kind of comprehensive look at how you combine these things. But certainly with a short interval, you get a lot of reactogenicity with your second dose of mRNA, and I'm sure people would recognize that, and less with the adeno. But as you extend the interval, your anti-vector immunity for the adeno drops, and so you can get more cytokines. So your reactogenicity goes up. But if you extend the interval for the Pfizer or Moderna, it's the same. Then your anti-spike immunity will wane, and therefore your reactogenicity goes down. So just extending the interval is just one simple way of modifying these quite significant reactogenicity events. And I think there'll be essentially a little bit more that you could play with these RNA vaccines to really get the best out of them. So that's that. And I'm going to show you now an example of tissue repair quickly. So what we found when we were doing these experiments in triggering the cells and doing transcriptional studies is that one of the features that comes out if you give TCR triggering is something that's just been described as tissue repair. It's a bunch of genes that have been associated with tissue repair as a sort of one of these gene signatures. And it's found in humans and mice, and lots of groups found it roughly the same time. And it's been shown in vivo as well that these cells can actually, in a mouse models, that these cells can actually provide wound healing, which isn't exactly the same as tissue repair, but certainly if you give a wound, these two papers showed that if you add in MAIT cells under different conditions, you can help the repair of it. And what we've been doing is in vitro, because we don't really have the wound healing models. We've done an in vitro wound healing model where we've added MAIT supernatants, triggered the MAITs with E. coli, and then blocked the TCR. And you get these sort of, you can measure basically the gap so these are sheets of transformed cell lines. You make a wound, it will heal up. If you add MAIT cell supernatants, it heals up slightly quicker. And if you block the TCR, you can block at least partially some of that. So that sort of supported the idea it could be important, but we didn't really know how. And so if we use, I'm going to just sort of shorten some of this, but if you just trigger the MAIT cells in vitro and take their supernatants, and we found that combined triggering, as I showed you, that gives you the maximum kick. So if you give TCR triggering and cytokine triggering, and then just take the supernatants and add it to those cells. Well, first up, we measured what was in those supernatants. And what was quite interesting is we got, well, there's lots and lots of things, but I'm just showing you the VEGF, the EGF, vascular endothelial growth factor, is actually quite well made by MAIT cells, amongst other things. There's a gamma and GM-CSF and so forth. But we were interested in the VEGF because that might have a wound healing role. And if you sort a whole bunch of cells, so not just MAITs, but regular CD8s and CD4s, you find that lots of cells can make GM-CSF and some of these other molecules, but it's only really the MAIT cells that are making, excuse me, it's only really the MAIT cells that are making apart from a few rogue naive CD4 cells here, which is kind of slightly annoying for the graph, but statistically, there's still quite a big difference. So that's pretty much nailed on. And then if you do the wound healing, that we got a bit better at doing this second time around. So we've got like an insert so that the wound is a bit more standardized. And you can sort of measure again the wound healing with the tissue culture treated TNC, T cell and cytokine treated MAIT cells, and you can see it heals up a bit better. Just MAITs on their own does something. So we think that they are involved in wound healing. And so the question is, how much of that is VEGF? And VEGF binds VEGF receptor 2. And there's lots of biology that's understood about this, but it's involved in many functions of the cell, but certainly migration and proliferation, which are the things we're really measuring. And so if we block VEGF receptor 2, you can actually abrogate all the wound healing. So that's with the treated MAIT cell supernatant, and that's with the blocking. So I think that's at least one of the things they do, and there seems to be something a little bit special about it. So it's sort of gone from thinking that it's a bit of a vagary to something the cells are really designed to do, and they'll do it well. And many other cells at least don't do it so easily. And it's true in the liver as well. So one of the questions, and I didn't think we'd be able to answer this, is whether this happens in vivo, but there's a nice experiment that's been published using a model where prior to liver surgery, a lobe of the liver is embolized. And the idea of that is that you get some regeneration of the remaining lobe, because the liver's got this fantastic capacity to regenerate. And that means that when you do the surgery to remove the embolized area, which has got your lesion in it, that's why you're having the surgery, the rest of the liver is in a better state to take over. And so a single cell study had been done by this group, and we extracted the data for MAIT cells. And you've got MAIT cells from different parts of the liver. So there's the embolized region, there's regenerating bits of it, and some healthy. So if you just compare the healthy and regenerating, the question is whether there was any VEGF found. And actually there is a bit, and it's not very many dots, but they're not very many MAIT cells, but it's pretty clear that at least we can find some transcripts for VEGF. There's different isoforms, but this is certainly an active one that you don't really see in unstimulated liver, but you can see in these regenerating livers. So I think there's at least some data suggest it's probably happening in vivo. But ultimately, we'll probably need to do some mouse model if you really want to show how it's really working. But actually, this is where things start to break down a bit, because they're not completely equivalent, the programs in humans and mice. So I showed that. So the last little bit, I just wanted to briefly mention other cells. So if you stimulate MAIT cells, as I showed you, with either cytokines or TCR, you can get these interferon gamma more with the cytokines, other functions with the TCR. And it's actually very, very similar. If you look at, these are delta 2 cells and these are MAIT cells, and this is very similar experiment to what I just showed you, and they're very similar profiles. So delta 2 cells, which are again commensal specific, different molecules and different mechanisms, but ultimately very similar roles. They look very similar actually. And so we were asking about CD4 cells. So if you take this view a bit further, we thought that some CD4 cells might also follow this pattern. So Philip was looking at E. coli, staph, and candida responsive cells in the gut, and he found that the cells which are reactive to these commensals were all really high in CD161. So we're looking at reactive cells by TNF production. And it's really interesting that they all have this feature which is high in CD161, just like the MAIT cells. But they're all conventional class 2 restricted cells. If you block class 2, you can kill off all of this reactivity. And if you block MR1 or cytokines, it doesn't really have the effect. So that is kind of the first feature. And we sort of divided the cells up according to the amount of CD161, and some of them also express CD56. And as you go up that gradient, they express this molecule PLZF, which is a really important transcription factor for driving some of this innate-like features. So you can see there's more in a MAIT cell, but there's not much in a regular cell. So that's really interesting. And if you trigger these cells, which are straight out of the colon, we get the same features as we just looked at before, that if you give them IL-12 and IL-18, they'll make gamma. So they don't need their TCR. So that's probably the PLZF that's driving that. And also, if you follow your way up the same gradient, you'll see that the cells also express raw gamma. And so you can predict a whole bunch of their features, R17 and so forth, that they'll make. So far, they look really like MAIT cells. It's just that they got a conventional TCR. And what's quite interesting, and this is what Philip's gone on to do, is that if you do single cell, they actually, this group of cells, which you call T-mix, so microbially reactive, innate-like, actually, now I've forgotten the C. I have to remember. It was not colon associated, but it's anyway, they're T-mix. They are distinct from other cells. So I think it's not, we just haven't invented these. I mean, this is sort of independent program. And they also, if you look at them as they're stimulated and look for their tissue repair signature, they are the cells that express more than the others. And as you increase the stimulation with the T cell receptor triggering, they'll make that sort of, those probably exactly the same set of factors. We haven't explored it yet, but at least at a transcriptional level, we've got the kind of left-hand side of the diagram. So the sort of model that's developing is sort of slightly gone slightly beyond what we originally thought was that it's a bit like an NK cell. They'll fire off when you get a virus or tissue damage, and they'll make lots of gamma, and that's more or less it, to something that's a bit more nuanced. So on a daily basis, I think a MAIT cell probably receiving some TCR triggering, and as far as we can tell, they'll make growth factors, things like VEGF maybe, or perhaps furin, other factors which just keep the epithelium attack, which is an important part of host defense, but one which we often neglect. But if the microbes end up the wrong side and there's tissue damage and inflammation, danger signals rather, the MAITs will help recruit other cells, and they'll perform their own functions, which is partly inflammation, but also partly later repair. So that's, I think we can say that for MAITs, and we know that MAITs sit just there. Like when we look in the colon, for example, they sit really abutting up to the epithelial cells, and I showed you the liver data. And I think T-mix have probably got a similar role. So these are CD4 cells which are microbially responsive, and probably on a daily basis they're not doing much other than just keeping things ticking over, but they can respond very strongly if there's inflammation, and we've shown that they can get involved in, for example, inflammatory bowel disease. So I think a very parallel sort of biology there, but many more questions about where they're getting their antigen and how they become T-mix, and essentially how that whole program is induced. So I think I'll stop there. Thank everybody, and thanks for having me.
Thanks, Paul. So if there's questions in the room, could you go to the microphone? And if there's questions online, can you put them in the Q&A section?
Thanks, Paul. Nice to meet you again. Hopefully Tirun is doing great in the lab. You're not kicking her butt too much. Two questions. One was about, I was intrigued by this phenomenon where MAIT cells takes time and to do what they do. But you said they can do Th17 response, but I kind of don't see them often doing it.
Yes.
Do you think is some sort of peripheral tolerance mechanisms that keeps those cells in check? Because they could do a lot of things, but they don't.
I don't think it's tolerance so much as they just need more signal. They seem to need more signal to get them to that threshold. I think there's, if we look at all of the things that we look at in terms of function, well, activation, granzyme B, interferon gamma, IL-17, they're all on a bit of a gradient. So to get to make CD69, you just need interferon alpha. To get them up the next phase, you probably need IL-12 and so forth. And the TNF in our model was needed to make the gamma. And I think there's an extra bit that's needed to make IL-17. So it's tolerant in some sense that they need a higher activation threshold to reach it. And part of that is, we think, opening up the chromatin around IL-17. But I don't think there's anything really suppressing them. And it may be that the signals don't have to be kind of contemporaneous, if you see what I mean. Once you got into tissue, let's say those commensal, the cells were just sitting there, they're getting a lot of TCR signaling anyway. So it's relatively easier for those ones to make IL-17 in the presence of cytokines, but they still won't make it until they get a good kick.
Oh, thanks. A second question related to that. So about this idea that like you are starting to see on CD4, but this kind of, we call it unconventional NK-like behavior is started to be published 10 times a month in pretty much any context where you have inflammation or immunodepletion, where there's a high perturbation of the environment.
Yes.
And we look at CD8 T cells, and they have not even the remembrance of that MAIT cells. They're absolutely not MAIT cells, but their single cell data and proteins looks completely identical to MAIT cells.
Yes.
And they're happening everywhere. So what do you think about that going back? So MAIT are always there to do things, but CD8 and CD4 can actually do exactly what MAIT cells do.
They can eventually. Now, I think the way I look at it is that the MAIT cell is essentially pre-prepared to do all this stuff, and the other cells can get there, but they need a lot more work, and they need to get into tissue first and receive the signals. But I don't think there's no unique thing that a MAIT cell can do. It can do a lot of everything. So I think the reason that we see a phenotype when we take them in and out is largely because they're there first. So if you're looking at something where timing is really important, then then you might see this sort of early effect. And it's not because they're doing something unique. It's just they're there at the right time in the right place. But I think you're right. And so yeah, in inflammatory diseases, you see a lot of TC17s, which you don't normally see, and so forth. And yeah, certainly in the mouse in particular, average memory cells will become much more cytokine dependent. Having said all of that, I think even in that context, they're still the most cytokine dependent cells. If you look at the work we've done in COVID, they're the sort of real indicators of inflammation. That they're right at the extreme end. Lots of other cells can sort of do it a bit, but they're kind of always a little bit ahead. Probably just because they've got that kind of inbuilt programming.
Great talk, thank you. I'm interested in the T-mix cells, so it's great to have another subset of T cells because there just aren't enough. But how are you defining T-mix? Like, are there phenotypic markers or is it
I didn't show all that because in the original study we defined them according to their responsiveness to microbes. They're microbe reactive, innate-like, and so those are high in CD161 and other markers, but the other markers weren't brilliantly defined. But because we've done all that single cell work, we've got some other markers which I think are a bit more tractable. So hopefully it'd be probably a combination of those, because the thing is to define them with a functional assay is really clunky, and you know, you need a lot of tissue. But I think now with Philip's probably latest iteration, we could define them. So galectin-3 seems to be a really good marker, and probably something like CCR2.
Okay. Are they 4s and 8s? 4s and 8s?
So those, no, we haven't found any. I think you might expect to find a CD8 maybe somewhere else that was also perhaps in the skin that's commensal reactive that had acquired this, but in the gut, it's these CD4s, and there are a lot of them. So I think it's not a small population. And one of the things was a bit of a problem is the phenotypically, you know, amongst those very high CD161, CD56 high cells, that's quite a big population. And I think when we add in those extra markers, that's pretty solid. But the numbers that are responding to the microbes that we tested, E. coli and so forth, is quite a small set. So I think if you use the function, it's a little bit, you know, you're missing a lot. Yeah. So I think we want to publish those features, so that would allow more people to do more experiments, and then you can really start to kind of dig into that.
And just secondly, why do you think the MAITs are so enriched in the liver if they're responding to commensals? Like, is that to do with, do you think there's that many commensals in the liver, or is it to do with the blood supply coming? It's the blood supply thing.
So we know that the ligand is present in portal vein blood, and it's actually very strongly loaded onto hepatocytes for some reason, that they're very easy to load if you compare them to other cells. So I think that's probably the reason.
Right.
Yeah.
Okay.
Hi, Paul. I was just interested in the, I guess, damage repair function of MAIT cells. Has there been a lot of work done in COVID, particularly in the respiratory tract, and thinking along the lines of long COVID, whether you think they'll play a role there?
Well, I don't know about long COVID and that repair. I think in what happened in what you can see in COVID is a massive drop in the cells in acute disease, which is typical for any acute disease. So I showed you those data from dengue and dengue and flu. So they're activated, but they're also depleted. So they're probably disappearing off into tissues. And you can find them in tissues, but it hasn't really been possible to sort of show them particularly localized according to pathology so far in any really meaningful way. So what we did find was that the activation, acutely at least, the activation seemed to be associated with poor outcomes. So as part of all that really bad cytokine milieu, which the ICU patients are sort of suffering from, and that you can block with anti-GM-CSF and anti-R6 and steroids and so forth, they're very much kind of involved in that. But then, as we were just discussing, so are a lot of other cells. So they're very good markers of it, but they're probably contributing to that pathology. I don't know whether at the end of all that, they could help with some of the healing up process. It'd be really interesting whether people's ability to kind of repair was influenced by their sort of MAIT cell functionality. But again, you know, there'd be one of many cells which have got that potential, especially once you've had a kind of really severe lung damage. So I would be surprised if they were the thing, but it's not, you know, not impossible. Yeah, we've had a look at some wild mice that they captured in the woods in one of the chicken houses, and they don't have any more MAIT cells. I was kind of disappointed. They don't have, what they actually have more, they have fewer iNKT cells as well. So in the liver, so what happens in mice is that if you look in the liver, the mouse liver's got a lot of iNKT cells in, whereas the human liver has a lot of MAIT cells in. They're sort of flipped and flopped. Was actually a wild caught mouse, had very few iNKT cells in its liver, but they hadn't been replaced by MAIT cells. So we don't really know what's going on there, but it's certainly not something you can't really right the wrong by just making it kind of having a normal microbiome.
Great. Well, I'll just, if there are no more questions, I'll apologize on Tony's behalf because he had to run away, got a call from the hospital. But to thank you very much again, Paul, for coming all this way and giving us a very interesting talk. So those online as well for joining us.